So we're doing a lab today that involves testing substances for the presence of molecules that are commonly found in foods. We're going to be testing these substances for lipids, which are fats, proteins, starches, sugars, and finally we're going to test the pH because, as we'll learn in our next lab, these are things that affect whether substances undergo chemical reactions during cooking. When you start the lab, make sure you follow the safety precautions. Some of the liquids that we use in this lab are dangerous to get in your eyes, and they're also not great to get on your skin. So if you get something on your skin, just wash your hands or skin right away and let me know. Don't get things in your eyes. You have splash goggles that you should wear anytime you're handling liquids in this class. If you do get something in your eyes, let me know right away because I will need to send you to the nurse. We're each going to be testing a different food substance, and there are enough materials for you to work individually at your tables. My food substance is labeled with number 4, and it's a liquid. Some of the substances are powders. Make sure that you record your data in the correct row in the chart. I'm going to make sure to record all my data next to number 4. The goal at the end of this experiment is that we can share our data with each other, and also that we can try to identify the food substance that you studied. So to begin, I'm going to take the empty beaker and place it on the scale. I'm going to zero the scale. Then I'm going to add 2 grams of my food substance. In this case, this substance is a liquid. So I'm just going to add until I see 2 grams on the scale. We zero the scale to make sure that the scale is ignoring the beaker and only measuring the substance that we're adding. As long as it's close to 2 grams, that would be fine. I just want to make sure you have enough and that we're not wasting materials. If your substance is a powder, then after you add the 2 grams of powder, you're going to add water from this container until it's a liquid substance. We want these to be able to be dripped to use in some of the tests. So if you can, add water from the water bottle until the substance that you have is more liquefied. Some will require more water than others. Part of the reason that we do that also is so we can observe whether it's soluble, does it dissolve easily in water, and record that in the chart. So if you have a liquid like I do, you can just add a couple of drops of water to that and try to notice if it mixes or if they stay separate. If you see different colors, layers, or bubbles, that would be a sign that it's not soluble. In this case, mine mixes easily, so I'm going to assume that this liquid is soluble in water. The next test is the lipid test, and I already started that one. All you do is take a couple of drops of your food substance and you place it on the brown paper bag. Then we want to wait for the brown paper bag to dry, and once it dries, we hold it up to the light to see if the food substance has affected the brown paper and made it more transparent. If it's transparent, the light will pass through it easily, but we want to let it dry first. The next test is the protein test. For this test, we're going to add 3 drops of our food solution to a well plate. And then we're going to add 2 drops of Biuret solution, which is an indicator used for proteins. We're making sure not to stick our pipette in the solution because that would contaminate it. Just let the drops fall. And for that one, we do need to wait about 20 minutes, so you might want to set a timer on your phone. The next test is the starch test, and it's similar. We're going to add 3 drops of food solution to the next well. And then we're going to add 2 drops of iodine, which also would be dangerous to get in your eyes, and you should avoid getting it on your skin. Iodine is a stain that's commonly used in life sciences. For both of these tests, what we're looking for is a kind of dramatic color change. So far, they look to be about the same color that they are in the bottle. We do need to wait about 20 minutes for the Biuret solution, but we can tell right away that that's negative for starch because the iodine did not change color. The next test requires heating, so you're going to take a test tube. If there's more than one group at your table, you can mark the test tube or try to notice how the test tubes are different so that you don't mix them up. I'm alone here, so I don't need to mark my test tube, but I do need to add 1 milliliter of my food solution to the test tube, and then 1 milliliter of Benedict solution. These pipettes are designed to measure a milliliter if you fill them to the line that's just below the bulb. So I'm going to squeeze the bulb while it's in the solution. I'm going to let it fill up to the line. When it gets to the line, I'm going to release the bulb and transfer it to the test tube, and I'm still trying to avoid contamination here. Notice that the Benedict solution is kind of a light aqua color. Again, we're looking for a color change, but this time after heating. So I'm going to place this test tube in the hot water bath, and I'm going to make sure that when I do that, I only handle the cold glass at the top of the test tube, and that I point it towards the wall and away from people's faces in the water bath. Set a timer for about 4 minutes and then check to see if there's any color change. If it seems that it's changed a little, you can let it go for another minute or two and then check it again to be sure that it was heated enough. The final test is a pH test, and for the pH test, you're just going to take a strip of pH paper from this container, and you're going to touch it to your original food substance that's in the beaker, and then you're going to compare it to the chart that's on the side of the pH paper. So in this case, sometimes it helps to put on a white background. I can tell that the pH paper is like a yellow color. And if I look at my pH chart, that tells me that this is a neutral substance. This is a clear, neutral substance that does not contain starch, does not contain proteins unless that changes color, does not contain sugar unless the Benedict solution changes color in the next few minutes. And I need to wait, and then I'm going to hold this up to the light to see if it contains lipids. If you're not sure, you can also record things like slight positive or positive with a question mark. Just be as detailed as you can so that we understand your results. At the end of that, if you need to do some research to find out what your food substance is, you can. I will also have some options projected that you can look for. Most importantly, be safe during this lab.